in this live chat. If I give you a tablet right now, can you look at that tablet and tell me whether it is really safe, effective, pure and manufactured correctly? Probably not. You may look at the color, you may look at the shape, you may look at the packaging, but can you identify whether the correct API was used? Can you tell whether the API quantity is correct? Can you tell whether the manufacturing process was under control? Can you tell whether the cleaning procedure was properly executed? Can you tell whether the equipment was calibrated? Can you tell whether the analyst followed the approved analytical procedure? Can you tell whether the data was genuine? You cannot. And that is exactly why CGMP exists. Today we are going to talk about something that is the backbone of pharmaceutical manufacturing. Current Good Manufacturing Practices, or CGMP. And I don't want this session to be just another definition based lecture. I want this to be practical. I want you to imagine that we are actually walking through a pharmaceutical manufacturing facility together. We will start from the moment raw material enters the plant. Then we will move through sampling, dispensing, manufacturing, in-process testing, packaging, quality control, quality assurance, documentation, deviation, OS, CAPA, change control, validation, data integrity, and finally we will reach the most important destination, the patient. Because ultimately CGMP is not about paperwork, it is not about passing an FDA inspection, it is not about making the auditor happy, it is not about having thousands of SOP. CGMP is about making sure that the medicine reaching the patient has the quality it is supposed to have. So if you are working in quality control, quality assurance, production, engineering, validation, regulatory affairs, warehouse, microbiology, analytical research and development, or any other pharmaceutical function, stay with me because I am going to explain CGMP from the practical industry point of view. And throughout this live, I will also ask you some interview style questions. So if you are preparing for a pharmaceutical interview, this session can be extremely useful. And if you are already working in the industry, I want you to compare these concepts with what you see in your own plant. Let's begin. Part 1. What exactly is CGMP? First question. What is CGMP? CGMP stands for Current Good Manufacturing Practices. Now notice the first word, current. Why is the word current important? Because pharmaceutical manufacturing cannot remain stuck in the past. Technology changes, equipment changes, analytical techniques changes, manufacturing process changes, risk understanding changes, regulatory expectation evolves. Therefore, pharmaceutical companies must maintain systems that remain appropriate and effective. FDA explained that the C in CGMP emphasizes the use of current technology and system appropriate for preventing contamination, mix-ups and errors. Now let's make it simple. Imagine two pharmaceutical plants. Plant A says, we have been doing this procedure for 20 years, so it must be correct. Plant B says, we have been doing this procedure for 20 years, but let's evaluate whether it remains scientifically justified, compliant, effective, and suitable for current technology. Which plant has the better CGMP mindset? Obviously, plant V. Because CGMP is not, we have always done it this way. CGMP is, we have a scientifically justified, controlled, documented, and effective way of doing it. That is a completely different mindset. Next part, why is CGMP so important? Let's take a very simple example. Suppose a manufacturing batch contains 1 million tablets. The laboratory may test a relatively small sample. Now imagine that the sample passes. Does the automatically prove that every tablet in the batch was manufactured correctly? No. And this is one of the most important concepts in pharmaceutical quality. Testing alone cannot build quality into a product. Testing can provide evidence about the tested sample. But quality must be designed and controlled throughout the manufacturing process. FDA specifically emphasizes that testing alone is not adequate to ensure drug quality because only samples from a batch are tested. So we have two concepts, quality by testing versus quality by design and control. Modern pharmaceutical quality strongly focuses on the second approach. Let me give you an example. Suppose your tablet assay specification is 95% to 105%. You test the finished product, the result is 99.5%. Excellent. But what if the wrong material was dispensed? What if the mixing time was incorrect? What if the blend was not uniform? What if the compression machine had a problem? What if the analytical balance was not calibrated? What if the sample was not representative? What if the analyst made a calculation error? What if the raw data was manipulated? A final number of 99.5% alone cannot answer all these questions. That is why CGMP is a system, not just a laboratory test. Next part, the pharmaceutical quality mindset. Now I want everyone watching this live to remember one sentence. Quality is everyone's responsibility. Quality is not only QA responsibility. Quality is not only QC responsibility. Quality is not only production responsibility. Quality begins with people. It continues with materials. It continues with equipment. It continues with process. It continues with documentation. And finally, it reaches the patient. Think about a chain. Raw material, dispensing, manufacturing, testing, release, packaging, storage, distribution, patient. If one important link fails, the final product can be affected. Therefore, CGMP is a chain of controls. Next part, 21 CFR, 210 and 211. Now let's come to the regulatory side. If you are preparing for a USFDA-related interview, you should know this. For finished pharmaceutical, two important regulations are 21 CFR Part 10 210 and 21 CFR Part 11. Part 210 deals with CGMP in manufacturing, processing, packaging or holding of drugs. Part 11 contains CGMP requirement for finished pharmaceutical. Now don't just memorize these numbers, understand them. If an interviewer asks what is 21 CFR Part 11, don't simply say finished pharmaceutical. Continue, say that it establishes CGMP requirements covering areas such as personnel, building and facilities, equipment, components, production and process controls, packaging and labeling, laboratory controls, record and other important quality systems. That answer demonstrates understanding rather than memorization. Next part, personal, the first line of defense. Let's start with people. Imagine a perfect pharmaceutical plant, modern HBAC, modern HPLC, modern manufacturing equipment, automated system, excellent laboratory, but the people are not properly trained. What happens? This system can still fail. Therefore, personnel are one of the most important components of CGMP. People must have appropriate education, training, experience, responsibilities, and appropriate understanding of the procedure they perform. Now think about a QC analyst. Can an analyst simply enter the laboratory and start operating an HPLC? No. The person needs appropriate training. Training may include SOP training, instrument training, analytical procedure training, safety training, data integrity training, good documentation practices, and where applicable, qualification or competency assessment. Here is a practical question. Suppose an analyst has worked on HPLC for 5 years. Does the automatically mean the analyst can perform every HPLC method in the laboratory? No. Experience is important, but training and authorization for the applicable procedure are also important. This is a common interview concept. Next part, buildings and facilities. Now let's walk into the facility. Why do pharmaceutical facilities require controlled environments? Because contamination and mix-up are major risks. Imagine two products being manufactured in the same facility. Product A contains an API, product B contains another API. If proper controls are absent, cross contamination can occur. Now imagine a highly potent compound. Even a small amount of unintended carryover can become a significant quality concern. Therefore, facility design is not simply about having a beautiful building. It is about risk control. Important consideration can include material flow, personal flow, air handling, segregation, cleaning, environmental controls, lighting, space, sanitation, waste movement, and prevention of mix-up. Ask yourself, why do we have separate area? Why do we control personal entry? Why do we control material movement? Why do we have status level? Why do we have line clearance? Every one of these control has a purpose. Prevent error before they become product defects. Next part, equipment. Now let's talk about equipment. Think about an HPLC. What do you need? Instrument qualification, calibration where applicable, maintenance, system suitability, appropriate software controls, data integrity controls, approved analytical procedure, trained analyst. Now think about manufacturing equipment. A blender, a granulator, a compression machine, a coating machine. Each piece of equipment must be suitable for its intended use. You cannot simply purchase equipment and say it is new, therefore it is qualified. No. New does not automatically mean qualified. The equipment must be appropriately evaluated and qualified according to the site's validation program and applicable requirements. Next part, calibration versus qualification. This is a very common interview question. What is the difference between calibration and qualification? Let's make it simple. Calibration is generally about stabilizing the relationship between a measurement device or instrument and an appropriate standard. Qualification is a broader. Qualification provides document evidence that equipment or system are suitable for their intended purpose. For example, an analytical valence may require calibration, but the laboratory equipment and associated system may also be subject to qualification activities. Similarly, manufacturing equipment may undergo DQ, IQ, OQ, and PQ depending on the system and applicable validation strategy. Remember, calibration asks, is the measurement accurate within the required criteria? Qualification asks more broadly, is the equipment or system suitable and capable of performing its intended function? Next part, raw material control. Now imagine a truck arriving at the pharmaceutical plant. Inside are raw materials. What happens next? Do we immediately use them? Absolutely not. The material must go through appropriate controls. Think about receipt identification, supplier controls, quarantine, sampling, testing or examination as applicable, status labeling, release or rejection, storage and traceability. A material should have a clear status. For example, quarantine, approved, rejected. The exact level and electronic status controls may vary by site, but the principle remains. You must know what the material is and whether it is authorized for use. Now imagine a warehouse operator accidentally picks a rejected material. That is a serious system failure. This is why physical segregation, status labeling, inventory controls, and computerized controls are so important. Next part, sampling. Now we reach a very important area for QC professional. Sampling. Sampling sounds simple, but scientifically sampling is extremely important. Suppose you have a container of raw material. You take one sample. Does the one sample necessarily represent the entire material? That depends on the sampling strategy. This sampling procedure must be scientifically justified and appropriately controlled. You need to consider things like number of containers, material characteristics, sampling locations, homogeneity, risk, historical performance, supplier controls, and applicable procedures. The sample must be representative because the laboratory result can only be meaningful as the sample being tested. Remember this sentence: a perfect analytical results from a non-representative sample can still lead to a wrong quality decision. That is a powerful interview statement. Next part, production and process controls. Now let's move into manufacturing. Suppose we are manufacturing tablets. We might have dispensing, sifting, granulation, drying, milling, blending, compression, coating, packaging. At each stage, control are required. Why? Because the final product quality depends on the process. Consider granulation. Important process parameter may include binder quantity, granulation time, impeller speed, chopper speed, endpoint criteria, moisture, depending on the product and validated process. Now consider compression. Parameter may include compression force, tablet weight, thickness, hardness, friability, disintegration, and other applicable controls. But here the important point, not every parameter automatically becomes a critical parameter. The classification should be scientifically justified and risk based. Next part, process validation. Now we come to one of the most important subject in pharmaceutical manufacturing, that is process validation. What is process validation? In simple word, it is documented evidence that a process operated within a stabilized parameter can consistently perform effectively and procedure a product meeting predetermined requirements and quality attributes. Think about making a cake. If you making one cake successfully, does that prove the process always work? No. You would want to understand ingredients, quantity, mixing, temperature, time, equipment, and reproducibility. Pharmaceutical manufacturing is much more controlled because the product is intended for patients. Therefore, process validation provides confidence that the process is capable of consistently producing an acceptable product. Validation versus verification. Another interview question. What is validation? What is verification? Validation is broader evidence that a process, method, system, or activity is capable of consistently achieving its intended purpose. Verification is generally checking or confirming that specified requirements have been met. The exact meaning depends on the context. So never give an oversimplified answer in an interview. Always explain based on the specific activity. Next part, cleaning validation. Now imagine product A was manufactured yesterday. Today we want to manufacture product V. What is the biggest concern? Carryover. Product A residue must not adversely affect product B. Therefore, cleaning procedure must be scientifically stabilized and appropriately validated or otherwise controlled according to the applicable system. Cleaning validation can involve selection of worst case products, identification of difficult to clean locations, acceptance criteria, sampling methods, swab sampling, rinse sampling, analytical methods, recovery studies, cleaning procedure effectiveness, and documented evidence. Now, here is a question, why do we perform recovery studies? Because if you swab a known surface and recover only part of the residue, the analytical results may underestimate the actual amount present. Therefore, recovery provides information about the ability of the sampling and analytical procedure to recover the analyte from the surface. This is where QC and validation work closely together. Next part, cross contamination. Let's imagine a facility manufacturing multiple products, product A, product B, product C. The biggest concern is not just contamination, it is uncontrolled contamination or cross contamination. Controls can include facility design, airflow controls, cleaning, equipment design, campaign manufacturing, dedicated equipment where justified, cleaning validation, personal practices, material flow, environmental controls, and appropriate procedural controls. The exact controls depend on product risk. For highly potent or sanitizing materials, the controls may need to be particularly stringent. The principle is identify the contamination risk, evaluate it, and establish effective controls. Documentation. Now I want to say something very important in pharmaceuticals. If it is not documented appropriately, it becomes very difficult to demonstrate that it happened. Documentation is not decoration, documentation is evidence. Suppose an operator says, I cleaned the equipment good, where is the record? Suppose an analyst says, I performed the analysis correctly, where is the raw data? Suppose production say, the batch was manufactured according to the procedure, where is the completed batch record? This is why documentation is critical. The documentation system provides traceability. Good documentation or GDP. Let's talk about GDP, good documentation practices. One simple principle is make the record accurate, clear, contemporaneous, and traceable. If you make a mistake in a paper record, don't use correction flow, don't erase it, don't make the original entry unreadable, follow the approved correction procedure. The correction should preserve the original information and provide appropriate traceability, including the reason where required by the applicable procedure. Now think about electronic records. The same principle applies. The system should maintain appropriate controls over user access, audit trails, data changes, electronic signatures, records, and system configuration. This brings us to one of the biggest topics in pharmaceutical quality, data integrity. Data integrity. Now listen carefully. You can have a beautiful facility, excellent equipment, highly qualified analyst, excellent SOPs, but if your data cannot be trusted, you have a serious quality problem. Data integrity means that data should remain reliable and trustworthy throughout its life cycle. A commonly used framework is ALCOA. A attributable, L legible, C contemporaneous, O original, A accurate. And modern data integrity expectation often discuss additional attributes such as complete, consistent, enduring, available. So ALCOA becomes ALCOA plus. Let's take an example. An analyst performs an HPLC injection. The analyst obtains a result. Later the analyst changes the result without appropriate justification or traceability. What is the issue? Data integrity. Another example. An analyst performs a test but record it much later from memory. What is the concern? Contemporaneous recording. Another example. Someone shares a login password. Why is that a problem? Attribution. You cannot reliably establish who performed the activity. Therefore, never share your login credentials. Data integrity is not just an IT topic, it is a quality topic, it is a patient safety topic. Quality control. Now let's talk about QC. Many people think QC means test and sample, but QC is much broader. QC activities can include sampling, testing, analytical procedure, instrument management, reference standard, reagent specification, stability testing, laboratory investigation, data review, and appropriate documentation. A QC laboratory is not simply a testing room, it is a controlled scientific environment. Specifications. What is specification? A specification defines the test, analytical procedure, and acceptance criteria with which a material or product must conform. Example can include assay, RS or related substance, dissolution, identification, water content, uniformity, microbiological attributes, physical characteristics depending on the product. Now remember, specification is not the same thing as process capability. A specification tell us what the product must meet. Process capability tell us how consistently the process performs relative to equipments. Both are important. Analytical method. Now let's talk about analytical methods. Suppose you have an HPLC method for assay. Can any analyst simply change the flow rate because the peak is taking too long? No. An approved analytical procedure must be followed. If a change is required, it must go through the appropriate scientific and quality system. The method may have characteristics such as specificity, accuracy, precision, linearity, range, LOD and LOQ, robustness, and post degradation depending on the method and intended use. ICH quality guidelines include Q2 for analytical validation and Q14 for analytical procedure development. System suitability. Now a common QC question. What is system suitability? System suitability is a set of tests used to demonstrate that the analytical system is performing adequately for the intended analysis. Depending on the method, parameters can include resolution, tailing theoretical plates, repeatability, retention time, signal to noise ratio, and other method specific criteria. But remember, system suitability is not a substitute for a validated analytical procedure, and passing system suitability does not give permission to ignore other requirements. OOS, out of specification. Now let's enter one of the most important area in QC, OOS. Suppose the specification for assay is 95% to 105%. Your result is 91.8%. What do? Do not immediately retest until you get a passing result. Do not simply discard the first result. Do not manipulate the chromatogram. Do not tell the analyst. Run it again and see if it passes. That can create a much bigger problem. Instead, the OS result must be handled according to the approved investigation procedure. The investigation should be scientifically sound and should determine whether there is an assignable laboratory cause or whether the investigation need to extend into manufacturing and other relevant areas. OOS versus OOT. Now another interview question. What is different between OOS and OOT? OOS means out of specification. The result does not meet the approved acceptance criteria. OOT means out of trend. The result may still meet specification but behave unexpectedly relative to historical or expected performance. For example, previous assay results 99.2, 99.5, 99.1, 99.4, 99.3, suddenly 95.4. It may still be within specification, but it could represent a trend that deserves evaluation. So OOS is a specification failure, OOT is a trend concern. They are not the same thing. Deviation. Now imagine something does not happen according to the approved procedure. Maybe the manufacturing time exceeded the approved range. A temperature went outside the defined limit. An instrument failed. A sample was missed. A procedure step was not performed correctly. What do? We initiate a deviation or appropriate quality events according to the site system. A deviation is not simply a form, it is an opportunity to understand what happened, why did it happen, what was affected, what is the product impact, what is the root cause, what corrective action is needed, what preventive action is needed. Root cause analysis. This is where many investigations become big. Someone makes an error. The investigation says operator finished. Is that a strong root cause? Usually not by itself. Why did the operator make the error? Was training inadequate? Was the SOP unclear? Was the procedure difficult to follow? Was there a workload problem? Was the interface confusing? Was supervision inadequate? Was there a system design issue? Was there a recurring weakness? Good investigation try to identify the actual underlying cause. Because if you only say retrain the operator, you may fix one person, but you may not fix the system. CAPA. Now we come to CAPA, corrective and preventive action. Corrective action address and identify problem or cause. Preventive thinking focuses on preventing recurrence or addressing potential systematic weakness depending on the quality system and terminology used. For example, problem repeated documentation error. B. CAPA retain the operator. Stronger CAPA may involve review the form, simplifying the procedure, improving system controls, adding verification, evaluating similar process, monitoring effectiveness. The keyword is effectiveness. Effectiveness, a CAPA is not truly successful just because it was close. We need to know, did the action actually solve the problem? Change control. Now imagine your company want to change an analytical method, a raw material supplier, a manufacturing parameter, an equipment model, a computerized system, a specification, a cleaning procedure. Can someone simply change it? No. Significant changes require appropriate change control according to the quality system. A proper change control procedure considers what is changing, why is it changing, what is the risk, what products are affected, what documents need revision, is validation required, is regulatory assessment required, is stability required, is training required, what is the implementation plan, and how will effectiveness be monitored. Risk management. Now let's discuss risk. Modern pharmaceutical quality use a risk-based approach. ICH-Q9 is the major guideline for quality risk management, while ICH-Q10 provides a model for a pharmaceutical quality system. Let's make risk simple. Imagine two activities. Activities A has a very low possibility of affecting product quality. Activity B has a high possibility of causing contamination of a critical product. Should both receive exactly the same level of attention? Not necessarily. Risk management helps us focus resources where the potential quality impact is greatest. Common risk management concepts include severity, occurrence, detectability, risk ranking, risk reduction, risk acceptance. The exact methodology may vary by organization. Quality by design. Now let's concept CGMP with a broader concept, quality by design, QBD. The basic philosophy is don't simply test quality into the product at the end. Build quality into development and manufacturing. Think about target product profile, critical quality attributes, critical material attributes, critical process parameters, risk assessment, design space, control strategy. This is a more scientific way of understanding pharmaceutical manufacturing. Instead of asking, did the final tablet pass? We also ask, why does the process consistently produce a tablet that passes? That is a much deeper quality question. ICH, Q8, Q9 and Q10. If you want to understand modern pharmaceutical quality, remember this group: Q8 pharmaceutical development, Q9 quality risk management, Q10 pharmaceutical quality system. Together, they provide a powerful quality framework. ICH Q10 describe a model for an effective pharmaceutical quality system and complete and com- and complements Q8 and Q9. So when you study CGMP, don't study regulation in isolation. Understand the relationship between development, risk management, quality systems, manufacturing, continuous improvement. Pharmaceutical quality system. Now let's ask, what is a pharmaceutical quality system? A pharmaceutical quality system is the overall system through which a pharmaceutical organization manages quality throughout the product life cycle. It includes things such as quality policy, management responsibility, documentation, training, deviation management, CAPA, change management, audit, risk management, knowledge management, continual improvement, and many other interconnected elements. The important word is system. A strong company does not depend on one excellent QA person. It has a system that consistently produces good decisions. Quality culture. Now I want to discuss something that is often ignored. Quality culture. Imagine an employee discover a mistake. If the employee thinks, if I report this, I will get punished. What happens? The employee may hide it. That is a dangerous. But if the culture says, report problem yearly, we will investigate scientifically and improve the system, then problems can be detected earlier. A strong quality culture encourages transparency, ownership, scientific thinking, speaking up, continuous improvement, and patient focus. Quality culture starts from senior management. Audits and inspections. Now let's talk about audits. A pharmaceutical company can face internal audits, supplier audits, customer audits, regulatory inspections. For example, FDA conducts inspection to assess compliance with CGMP requirements, but here is an important mindset. You should not manufacture quality only because an auditor is coming. If your system becomes compliant only 1 week before an inspection, you don't have a mature quality system. A good site should operate compliantly every day. An inspection should simply observe the system that normally exists. What does an FDA inspector look for? Imagine an inspector walks into your facility. What can they be interested in? They may look at personnel, training, facilities, equipment, production, laboratory controls, documentation, investigation, CAPA, change control, data integrity, or records, and whether your actual practices match your approved procedure. This last point is critical. Suppose your SOPs say step 1, step 2, and step 3, but operators actually perform step 1, step 3, and step 4. He missed step 3. You have a disconnect. That is a major quality concern. Therefore, say what you do. Do what you say. Document what you do. Review whether what you do remains effective. SOPs. Let's talk about SOPs. Standard operating procedures. Why do we need SOPs? Because important activities should be standardized and controlled. A good SOP should be clear, controlled, approved, current, accessible to authorized personnel, and understandable to the people who use it. But here is a common mistake. Companies sometimes create extremely complicated SOPs. The operator reads the SOP and still does not understand what to do. That is not effective quality. The best SOP is not necessarily the longest SOP. It is the SOP that clearly and consistently communicates the required process. Batch manufacturing record or BMR. Now imagine we are manufacturing batch number ABC123. The batch record provides documented evidence of what happened during manufacturing. It may include product information, batch number, materials, quantities, equipment, process step, parameters, in process check, operators entries, verification, yield, reconciliation, and other applicable information. Why is this important? Because months later, if there is a complaint, we may need to reconstruct what happened. This is called traceability. Batch release. Now the batch is manufactured, the laboratory completes testing, the results meet specification. Can anyone immediately ship the batch? Not necessarily. There must be an appropriate quality review and release process. Release is a quality decision. It should consider the applicable documentation and quality information. This can include manufacturing records, laboratory results, deviations, OS investigations, change controls, environmental or microbiological information where applicable, and other relevant quality events. The exact release process depends on the company quality system and applicable regulatory requirements. Stability. Now let's talk about stability. Suppose a product pass all release tests today. Does that prove it will remain within specification for its entire shelf life? No. That is why stability studies are important. Stability programs help evaluate how product quality change over time under specified conditions. We may monitor assay, degradation products, dissolutions, appearance, water, microbiology, physical properties, and other relevant attributes. Stability is essentially asking, will this product remain suitable throughout its labeled storage period? Complaints. Now imagine a patient reports these tablets look different, or the bottle was damaged, or the product did not behave as expected. What happens? The complaint enters the quality system. It must be appropriately assessed. Some complaints may be minor, others may indicate a serious quality issue. The company may need to evaluate batch records, testing, return sample, distribution, manufacturing history, previous complaints, and other relevant information. The purpose is not simply to close the complaint. The purpose is to understand whether there is a product quality issue and whether further action is required. Recall. In serious situation, a product recall may be necessary. Recall is one of the most important example of why traceability matters. If you don't know which batch were affected, where they were distributed, which customers received them, you cannot effectively control the problem. So the quality system must provide traceability from materials, manufacturing, packaging, distribution. Data integrity. Practical QC example. Let's take a very practical HPLC example. You are testing related substance test. You inject the sample. The chromatograms show an unexpected peak. What should you do? First, do not panic. Second, do not delay the injection. Third, do not manipulate the data. Fourth, follow the approved procedure. Evaluate the data scientifically. If an unexpected result or events meet the criteria for an investigation, follow the applicable investigation procedures. Now imagine the analyst thinks, I don't want an investigation, so I will simply delete this injection. This is exactly the wrong mindset. The correction mindset is unexpected data is information. Let's understand what happened. Laboratory investigation. When a laboratory issue occurs, possible areas of evaluation may include instrument, system suitability, standard preparation, sample preparation, reagents, mobile phase, glassware, calculations, analytical procedure, instrument maintenance, analyst training, reference standard, environmental conditions, and raw data. But remember, the investigation should be scientifically justified. You don't investigate randomly. You don't keep testing until something passes. You investigate based on evidence. Retesting and resampling. This is a very important topic. Retesting is not simply repeating a test because you don't like the result. Retesting and resampling must be scientifically justified and controlled under the applicable procedure. For example, if a documented assignable laboratory error is identified, appropriate action may be taken according to the investigation procedure. But if no laboratory error is identified, the investigation may need to consider broader manufacturing and product related factors. The key principle is never use testing as a tool to make an unwanted result disappear. Deviations, OS, OOT, CAPA, and change control. Let's connect all four. Deviation, something happened differently from the approved process. OS, a result failed on approved specification. OOT, a result may show an unexpected trend even though it remains within specification. CAPA, actions are taken to correct and prevent recurrence or address systematic weakness. Change control. A planned change is evaluated and controlled before implementation. These are not isolated systems. They interact. For example, a deviation may lead to an investigation. The investigation may identify root cause. Root cause may require CAPA. CAPA may require a change control. Change control may require validation. Validation may require training. And effectiveness may be monitored. That is how a mature pharmaceutical quality system works. Validation master plan. Now let's briefly discuss the validation master plan. A BMP provides a structured overview of the site's validation strategy and activities. It may cover areas such as facility qualification, equipment qualification, process validation, cleaning validation, analytical method validation, computerized system validation, utilities, and other applicable activities. The exact content depends on the organization and validation program. The important point is validation should be planned, controlled, documented, and scientifically justified. Computerized systems. Modern pharmaceutical manufacturing is highly computerized. Think about LIMS, ERP, MES, SCADA, HPLC softwares like Empower 3, Chromeleon, and Lab Solutions, electronic batches records, electronic document management system. These systems create and manage important data. Therefore, computerized systems require appropriate controls. Consider user access, audit trails, data backup, system security, change control, validation, periodic review, electronic signature, data retention. The goal is simple. The electronic records must be trustworthy. Audit trail. Let's take HPLC software. An analyst processes chromatographic data. Later, a reviewer checks the records. What if the software allows change to processing parameters? How do we note what happened? This is where the audit trail can be important. An audit trail can provide information about relevant system activities and changes depending on the system. It supports traceability, but remember, having an audit trail does not automatically mean your system is data integrity compliant. You also need appropriate review, controls, access management, procedures, and governance. Supplier quality. Now let's go back to raw material. Suppose your API supplier has excellent certificates. Does that mean you can completely ignore supplier qualification? No. Supplier quality is an important part of the overall pharmaceutical quality system. You need appropriate controls for supplier qualification, material specification, quality agreements where applicable, supplier performance changes, audit where justified, incoming materials controls, and ongoing monitoring. Because your final product quality can depend on your supplier performance. Quality agreements. When companies work the contractors or suppliers, responsibility need to be clear. Who performs testing? Who investigates deviations? Who manages changes? Who handle complaints? Who manage stability? Who reports quality issues? A quality agreement can define responsibilities between parties where appropriate. The important concept is responsibility must be clear. You cannot have a situation where both companies believe the other company is responsible. Contract manufacturing. Now imagine company A owns the product. Company B manufactures it. Who is responsible for quality? The answer is not company B. Quality responsibility must be appropriately defined and the product owner retains appropriately oversight consistent with applicable requirements. Contract manufacturing does not mean outsource the responsibility. It means outsource an activity while maintaining appropriate quality oversights. Training. Let's return to training. Training should not be a tick box activity. Imagine an employee completes an SOP training. They sign training completed, but they do not understand the procedure. Is the training effective? No. Effective training provides appropriate understanding and where required demonstrate competency. For critical activities, competency assessment may be especially important. Training should also be maintained when SOPs change, equipment change, method change, regulatory expectation change, responsibility change. Line clearance. Now let's talk about packaging. Imagine you have product A, you finish packaging product A, then you start product B. Ban prevent product A level from accidentally entering the product B packaging line. Line clearance and packaging controls. Before starting a new batch or product, appropriate checks are performed to ensure the area and equipment are appropriately cleared and ready. This is one of the most practical examples of CGMp preventing mix-ups. One wrong labels can create a serious patient risk. Level control. Think about a medicine level. information does it contains? Product name, strength, batch number, manufacturing information, expiry information, storage information, and other applicable information. A labeling mix-up can be extremely serious. Therefore, label issuance, reconciliation, storage, destruction, and line controls are critical. CGMP is not only about chemistry, sometimes a simple label error can create a major patient safety problem. Packaging reconciliations. Suppose you issue 10,000 labels. After packaging, you have 9,950 used, 20 unused, 5 damaged. What happened to the remaining 25? You need reconciliation according to the applicable procedure. Why? Because uncontrolled labels create a mix-up risk. This is why packaging controls are so important. Environmental monitoring. Now let's enter a sterile manufacturing environment. The risk level changes significantly. Microbial contamination becomes a major concern. Therefore, environmental monitoring can include appropriate monitoring of air, surfaces, personal practices, microorganisms, and other environmental attributes depending on the operation. The purpose is not simply to collect numbers, the purpose is to understand the state of environmental controls. Trending is important. One result may tell you something, a trend may tell you much more. Aseptic processing. For sterile product, contamination control is critical. Aseptic processing required carefully controlled personal behavior, material movement, equipment, cleaning, disinfection, environmental conditions, sterilization, or aseptic controls, process simulation, where applicable, and many other controls. The important mindset is sterility cannot simply be tested into a product after manufacturing. You need a controlled process that minimizes contamination risk. Microbiology. Microbiology is another major pillar of pharmaceutical quality. Depending on the product, microbiological controls may involve bioorder, microbial limits, sterility, endotoxins, environmental monitoring, water systems, and other applicable tests. The specific testing strategy depends on the product type and regulatory requirements. The key concept is microbiological quality must be controlled throughout the process, not just tested at the end. Water system. Let's talk about pharmaceutical water. Water can be a major raw material or process component. Therefore, water system require appropriate design, qualification, monitoring, sanitation, sampling, testing, trending, maintenance, and investigation of abnormal results. Depending on the intended use, different water quantities may be required. The key point is water is not simply water in pharmaceutical manufacturing. Its quality must be appropriate for its intended use. Utilities. Other utilities can also affect product quality. For example, compressed air, gases, steam, HVAC, electrical system, process gases. Utilities must be appropriately designed, monitored, maintained, and qualified where applicable. Again, the question is not is the utility running? The question is the utility capable of consistently supporting the intended pharmaceutical process without compromising quality? Continuous improvement. Now let's talk about improvement or continuous improvement. A mature pharmaceutical company does not say, we passed the inspection, so we are finished. Instead, what can be improved? Maybe reduce deviations, reduce recurring OS, improve process capability, improve laboratory efficiency, reduce manual error, improve data integrity, improve training, improve equipment reliability, im- improve supplier performance, that is continuous improvement. ICH-Q10 specifically provides a pharmaceutical quality system models that support continual improvement throughout the product life cycle. A real life style scenario. Now I want to give you a complete scenario. Imagine we are manufacturing a tablet batch. Raw material arrives, warehouse receives it, material goes into quarantine, sampling is performed, QC testing is completed, material is approved, production dispense the material, the batch is granulated, granules are dried, the material is milled, blending is performed, compression is start, in process checks are performed, tablet pass, coating is completed, finished product testing is performed, QC results meets specification, QA review the batch documentation, a deviation is identified, investigation is completed or closed, impact is assessed, CAPA is implemented if required, batch documentation is reviewed, the batch is released, the product is packed, the product is distributed. Now ask yourself, where was CGMP involved? The answer is everywhere, from warehouse receipt to patient. The most important CGMP principle. If I had to summarize the entire 1 hour session in one sentence, I would say build quality into the process, control the process, document the process, investigate failures scientifically, and continuously improve the system. Remember again, build quality into the process, control the process, document the process, investigate failures scientifically, and continuously improve the system. That is CGMP thinking. Top 15 CGMP interview questions. Now let's do a rapid fire interview section. Question number 1. What is CGMP? Current good manufacturing practice, requirements and practices designed to ensure pharmaceutical products are consistently manufactured and controlled to appropriate quality standard. Question 2. Why is CGMP important? To help ensure medicine are safe, effective and meet stabilized quality requirements. Question 3. What are 21 CFR Part 10 and 11 USFDA CGMP regulation for drug manufacturing and finished pharmaceuticals? Question 4. What is the difference between QA and QC? QC focus heavily on testing and laboratory controls, while QA provides broader quality system oversight and assurance. Question 5. What is OS? Out of specification, a result that does not meet an approved acceptance criteria. A result that does not meet an approved acceptance criteria. Question number 6. What is OOT? Out of trend, a result or pattern that may be unexpectedly different from historical or expected performance, even though it may remain within specification. Question number 7. What is deviation? An event or departure from an approved procedure, requirement or accepted process that required appropriate assessment under the quality system. Question 8. What is CAPA? Corrective and preventive actions designed to address identified problem and prevent recurrence of systematic weakness. I am repeating again, what is CAPA? Corrective and preventive action designed to address identified problems and prevent recurrence or systematic weakness. Question 9. What is change control? A controlled process for evaluating, approving, implementing, and documenting planned changes. Question 10. What is validation? Documented evidence that a process, method, system, or activity is capable of consistently achieving its intended purpose as applicable. Question 11. What is calibration? Comparison or adjustment of a measuring instrument against an appropriate standard to stabilize measurement performance within a specified requirement. I am repeating again, what is calibration? Comparison or adjustment of a measuring instrument against an appropriate standard to stabilize measurement performance within specified requirements. Question 12. What is data integrity? Maintaining trustworthy, reliable, complete, and traceable data throughout its life cycle. Question 13. What is ALCOA? Attributable, legible, contemporaneous, original, and accurate. Question 14. Why is training important? Because people performing GMP activities must have appropriate knowledge, training, and competence. Question 15. What is the ultimate objective of CGMP? Protecting the patient by ensuring consistent pharmaceutical product quality. Rapid fire live questions for your audience.